we're talking about NOD receptor involvement in Crohn's disease. Fellow immunology students, let's discuss a case study. Here's Bob, a 24-year-old male who came into the clinic for on and off rectal bleeding, weight loss, and diarrhea. After a blood test and colonoscopy, he was diagnosed with Crohn's disease. Crohn's disease is a type of inflammatory bowel disease, or IBD, where there's chronic inflammation of the GI tract. Crohn's disease affects around 500,000 people in America, both men and women equally, and can appear at any age. In terms of symptoms, the patients will often experience periods of severe symptoms called flare-ups rather than consistent symptoms. Unfortunately, the cause of Crohn's is unknown, but it has been established as a heterogeneous disorder where it is affected by multiple factors. With this, many factors have been identified to increase the risk or predispose an individual to developing Crohn's, such as smoking, genetics, and autoimmune diseases. Specifically, many genetic polymorphisms have been found to predispose an individual, where the main few, such as mutations in the NOD2 gene, are involved in the immune system. Today, we will be studying NOD receptors and how mutations in these genes may affect the appearance of Crohn's disease.
Let's get into node receptors because they play a significant role in the host innate immune system. Node receptors are part of the nucleotide oligomerization domain-like receptors. These intracellular receptors detect PAMPs as well as DAMPs, specifically for intracellular bacterial infections. Node receptors are similar in structure to TLRs. They have an N-terminal CAR domain, which is a distinct protein-to-protein association domain responsible for activating a downstream signaling pathway upon ligand binding. We also have a central nucleotide binding domain, which is involved in the oligomerization and activation of the receptor. And finally, they have a C-terminal LRR, which is what allows for the recognition of PAMPs and DAMPs. Node receptors exist in an active form in the cytoplasm. However, once the node receptor binds its bacterial ligand, it activates a cascading signaling pathway, essentially activating NF-kappa B, leading to cytokine production. These pro-inflammatory cytokines such as IL-1-B and CXCL8 play a key role in the recruitment of lymphocytes and leukocytes to the site of infection to kill the bacteria. There are three main types of node receptors, Node 1, Node 2, and NLRP3, but we'll focus on Node 1 and Node 2 in this case. Node 1 receptors are found in several types and are specific for fragments of peptidoglycan in gram-negative bacteria. While Node 1 has one CAR domain, Node 2 has two CAR domains, hence the respective names. The Node 2 receptor is an intracellular sensor for MDP, a small peptide that is derived from peptidoglycan that is found in both gram-positive and negative bacteria. Node 2 are found in phagocytes and pannid cells of the small intestine, allowing them to contribute to mucosal immunity. In the gut, Node 2 receptors have a role in regulating antimicrobial peptides such as alpha and beta defensins. It is the loss of function of Node 2 that is associated with the development of Crohn's disease.
We explore NOD2's role in Crohn's disease. Here's some background on the intestine and the gut microbiota. The human intestinal tract is colonized by many microorganisms, collectively called the gut microbiota. Some of these bacteria exert beneficial effects like helping nutrient metabolism, while others are pathogenic. A properly functioning immune system prevents the colonization of pathogenic bacteria and maintains tolerance to normal, also known as commensal, bacteria. The main bacterial sensor in the intestine is the NOD2 receptor, expressed in pannid and dendritic cells.
Since NOD2 receptor function is important, as mutations associated with NOD2 can lead to diseases such as Crohn's. These mutations will lead to lower TNF-alpha secretions, limiting the immune response to commensal bacteria. These mutations are normally found on chromosome 16, which affects the structural component CARD15, which recognizes LPS and activates NF-kappa B. Most of these mutations are frameshift, inserting C nucleotides causing premature stop codons in the LRR domain. Other mutations include missense mutations. The overall effect of this is the alteration of the LPS binding domain, which is the LRR or its adjacent structures, disrupting the function of the receptors, leading to a decrease in LPS activation, resulting in altered recognition or overactivation of NF-kappa B in monocytes.
So, loss of NOD2 function results in the inflammation and damage of the intestinal epithelia as seen in Crohn's disease. Many mechanisms have been proposed to explain how NOD2 mutations lead to Crohn's disease, two of which will be highlighted in this presentation. First, failure of NOD2 to respond to MDP results in the impaired secretion of antimicrobial compounds like alpha defensins and TNF-alpha, which may allow certain pathogenic strains of bacteria to colonize the gut. For example, NOD2 usually recognizes E. coli, which can multiply intracellularly in Paneth and dendritic cells. However, without NOD2, the NF-kappa B pathway is inhibited and transcription of antimicrobial compounds does not occur, and E. coli, a pathogenic bacteria, multiplies rapidly. E. coli is known to disrupt the tight junction barrier in the intestinal epithelia. Overgrowth of pathogenic strains due to NOD2 deficiency also compromises the mucus barrier and leads to increased interaction between the gut microbiota and immune cells, which may lead to chronic inflammation associated with Crohn's disease.
NOD2 can activate the adaptive immune system through cytokine secretion, directing the development of T cell subsets, hence postulating that NOD2 mutations prevent the development of T regulatory cells. In a recent study, NOD2's role in tolerance to commensal bacteria was examined. Normal activation of NOD2 in the intestinal DCs of mice results in cytokine production of GM-CSF, which promotes Treg cell development. Treg cells maintain immune homeostasis at the gut barrier, including inhibiting T effector cell function, hence preventing the recruitment of additional inflammatory cells to the intestinal barrier. In the same study, mice with NOD2 mutations had fewer levels of Treg cells, suggesting a dysregulation between tolerogenic and pro-inflammatory immune responses in Crohn's. As shown in the diagram, loss of NOD2 function leads to altered bacterial recognition by DCs. When they enter the lymph node, they allow for the development of T effector cells but low levels of Treg cells. T effector cells recruit neutrophils, natural killer cells, and other inflammatory cells which release cytokines, creating a pro-inflammatory environment in the intestine. Without Treg cells, chronic inflammation develops, which leads to a damaged mucus and epithelial barrier in the intestine.
Unfortunately, there currently is no cure for this disease, but there are treatments to ease symptoms as well as reduce the number of flare-ups such as immunomodulators which act to calm inflammation. Further research is needed to understand if NOD2 can be a genetic target to treat Crohn's disease. Sorry, Bob.